Hello, and welcome back. We're now going to kind of shift focus a little bit, and we're going to talk about cloning in the context of therapeutic cloning, okay? Which is going to be a little bit different than reproductive cloning. Reproductive cloning, the goal of reproductive cloning is to generate a whole organism, all right? We're going to have the same process to generate cells that can be used therapeutically. We're going to use that nuclear transplantation. We're going to take the nucleus from one cell. We are going to insert it into an unfertilized egg, give it that electrical shock, get things to undergo cell division until it gets to the blastocyst stage. And then this is where the difference comes in, okay? Is those cells are going to be continued. That word should be continued. I'm apparently making up my own words now. Okay. So we'll just erase that. So the cells are continued in cell culture. So essentially what happens is those cells are grown in a petri dish. And you'll have to pardon my art here. It's not going to be fabulous. But this here is going to be our petri dish. Okay? And what happens, those blastocysts are going to essentially spread out on the cell, on the surface of the dish. And what you're generating out of this process are embryonic stem cells. All right? And these are considered embryonic stem cells because they did come from a blastocyst. And that blastocyst has the potential to become an organism. So therapeutic cloning still has some controversy associated with it. There's some ethical considerations that go along with therapeutic cloning as well as reproductive cloning. But what we're talking about with therapeutic cloning are the potential applications. Because when you have a cell that is an embryonic stem cell, or if we go with stem cell in general, a stem cell is an undifferentiated type of cell, okay? And stem cells can either be totipotent or pluripotent, okay? Totipotent stem cells can give rise to any cell type. And that's, in terms of embryonic stem cells, one of the advantages is that they are still considered totipotent. They could give rise to a liver cell, a heart cell, a neuronal cell, a pancreatic cell. The options are endless as long as we, as a scientific community, know what cues to give that cell in order to get it to differentiate to the cell type that is desired. So totipotent can give rise to any cell type. Pluripotent has a limited number of cell types that can be derived from that particular type of stem cell. So there's a big thing about freezing umbilical cord blood, because in umbilical cord blood there are some pluripotent stem cells that can give rise to any of the cells that make up the hematopoietic lineage. And when I say a big word like hematopoietic, hematopoietic means blood cells, okay? So the value in banking cord blood is if that child ends up having a disease related to the hematopoietic system. So if they end up with a leukemia, you have the stem cells that can be transplanted back in, and you're not going to have rejection of those cells because it's the person's own cells, okay? But pluripotent, therapeutically, less valuable because it doesn't give rise to any cell type. So you couldn't take those umbilical cord cell blood stem cells and have it be able to generate neuronal cells, for example, okay? Totipotent would have that advantage, okay? So in terms of therapeutics, okay, the applications toward health are things like folks with that's an I, sorry, Parkinson's disease. This is why Michael J. Fox is a big proponent of stem cell research, because those totipotent stem cells, having the ability to give rise to new neural tissues, might offer a new therapeutic treatment for people who have Parkinson's disease, okay? Now, in terms of stem cell research, it's been very controversial because of the source of the stem cell. And there is a ban for a good while on doing any more research related to embryonic stem cells, all right? And I think embryonic stem cells do have some potential towards therapeutics. I do also think there are some ethical issues to address there. What came out of that ban is that it turns out in some tissues, in the adult, there remains an undifferentiated cell in and amongst the differentiated cells. So adult stem cells have been identified, but those adult stem cells are again going to fall into that category of being pluripotent. So they're not going to give rise to every single cell type, all right? So in terms of genetic cloning and nuclear transplantation, the advantage that those embryonic stem cells have is you can take a cell from an individual, put it into an unfertilized egg, let it develop, isolate the embryonic stem cells, then those stem cells would be specific for that person that the cell was isolated from, all right? And I'll use Michael J. Fox as an example. All right, so if they took a cell from Michael J. Fox, created embryonic stem cells, those embryonic stem cells then could be transplanted back in to potentially help treat his Parkinson's disease. Because with Parkinson's disease, you get certain neurons that produce certain neurotransmitters that die off. So if you replace that cell type, you're going to make that neurotransmitter, the person's symptoms associated with that particular disease would then theoretically subside, okay? So that's the therapeutic advantage, all right? But again, with cloning, because of the source of those cells, there are still some ethical issues to consider with that, okay? So if you have additional questions about this process or how this works, please let me know.